Hello, this is Kelly Eversole, and I'd like to welcome you to a Phytobiomes Alliance informational webinar. And today we have with us Meredith Ashby from Biosciences, who's going to be talking about unbiased, efficient characterization of metagenome functions with PacBio HiFi sequencing. But before we get started, I'd like to kind of explain a little bit about what phytobiomes are and about the Phytobiomes Alliance.
Kelly, can you share your screen?
Oh, you can't see the screen.
Under the sharing tab, there should be a big play button. There we go.
Is it on now? Yeah.
Yep. Beautiful.
Okay. So we are a nonprofit consortium of industry, academic, and governmental scientists. And our goal is to build a foundation for site-specific phytobiome-based enhancement of sustainable food, feed, and fiber production. Within that, a lot of people have been confused about what we mean by phytobiomes or made assumptions. But in essence, we think of plant-based agriculture as a very complex system. It is a phytobiome. And by that, we really mean a plant in a site-specific environment, the plant in a particular area. And that includes all of the microbiome and the various microbiomes, the plant, as well as the soil microbiomes, all of the microorganisms within that, as well as all the macroorganisms and macrofauna. It also includes the arthropods, other animals and plants that may be in that specific environment, as well as climate, weather, and water, and even water that might be added, such as irrigation, for example. And for us to begin to understand that entire complex system, we also have to take into consideration management practices, which would include nutrients, might include biologicals that may be added to beef up the health of the soil or to accelerate the development of the plant or even to increase the uptake of nitrogen in the plant. So within the Phytobiomes Alliance, our vision is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for a specific field in a given year. And to get there, we need to pull all of these components together and increase our understanding of the entire phytobiome system and really build up for next generation precision agriculture. And within those, if you think of these four balloons, so to speak, that are feeding into our knowledge of phytobiomes, we have the macroorganisms, which we have quite a bit of understanding of what plant genetics are doing and how they're impacting productivity. We also have quite a bit of knowledge about general management practices, whether that be crop rotation practices, nutrition, nutrient additions, or even soil amendments, etc. We also have quite a bit of information and knowledge about the physical environment itself, where we've got almost 70 years of weather data on a number of different levels that is available. Where we don't have a lot of information because the science has not been really robust enough or cost effective enough for us to get information is in the area of the microbiomes and really beginning to understand that. So that's become really a focus point for the Phytobiomes Alliance. And one of the main reasons that we are holding this webinar is that we'd like to see increased investment and increased research in the area of the microbiome. So with that, I'd like to introduce Meredith Ashby, who's the Director of Market Strategy for Microbial Genomics at Pacific Bio. At Caltech, mapping the gene regulatory networks that direct sea urchin development, just as the sea urchin genome was being sequenced. She was inspired by the impact of genome sequencing on her research and therefore joined PacBio after a postdoc at University of California, San Francisco. She's been with PacBio since 2009 and has worked in R&D on both sequencing optimization and bioinformatics application development. And right now she's focused on facilitating the use of PacBio long-read sequencing to solve the unmet needs of scientists seeking to understand the incredibly diverse range of microbes that live in, on, and around us in the systems we depend on. So with that, I'm going to turn it over to Meredith, and she will give you a short presentation about what PacBio can do.
Hi, Kelly. Thanks so much for that really kind introduction. I think I myself just learned quite a lot about phytobiomes listening to your description of the challenge. Can you see my screen?
Yes.
Great. Okay, so as Kelly noted, I'm here to tell you about what PacBio can do for researchers who are interested in getting a deeper understanding of metagenomes in particular. This is an exciting field, and until recently, there has not been a lot of progress. But with the changes in the throughput of both NGS and long-read technologies, we're starting to see a lot more insights coming forward. But there's still quite a few challenges that remain. And I think the biggest one can be summarized as, so far there's been a lot of sequencing, but there's not a lot of fully defined metabolic pathways. And this is true both for human microbiomes, but also particular for environmental samples as well. And what are the things that are really slowing our progress? So one of the big challenges that researchers face is that DNA extraction methods introduce bias. The differences between gram-positive and gram-negative bacteria and what is required to open them up is very well known. And another challenge is that people might not be so aware of is the challenge of assembly. So right now, most efforts to understand microbiome or metagenome data depend on assembly, either to increase the contiguity to the point where you can do gene binding, or for error correction, such that you can correct the errors by making consensus sequencing. And both of these methods are very data inefficient. And I think a statistic that really drove that home for me was understanding that with typical short-read metagenome assembly, anywhere between 30 to as high as 70% of the data cannot be backmapped to the assembly that's made. And that's simply because of the inefficiency of this process. And as it happens, the more complex your system, the higher the loss of data. So one question that people have been asking is, do we have all the data types that we really need to understand metagenomes? And people are answering that in a number of ways. So for one solution I've seen recently is people looking at spatial aspects of metagenomes, trying to retain that spatial information such that they can better understand which microbes interact with each other within a system. Another direction that has been fruitful recently is metabolomics or proteomics, looking at what compounds are actively being made so that you can understand what parts of the metagenome are under active expression and use. And then finally, of course, there's long-read sequencing. And since I'm from PacBio, you can probably guess which one of those new data types I'm going to be talking to you about. So with PacBio, we have our most recent system. It's called the Sequel system. And this is a very high throughput system that allows high resolution of metagenome data. And we have 3 different applications that you can run on the Sequel system, or at least we like to think of it as being 3 different applications. The first, of course, is full-length 16S sequencing. Because of our long reads, we don't have to choose which region of the 16S gene we want to sequence. You can sequence the whole thing. And the key utility of full-length 16S is to identify what species are present. And to use this approach on the PacBio system, you can either use our protocol, or you can make use of an all-in-one kit from our partner Shoreline Biome. A second application is HiFi shotgun profiling. And so this is it's just shotgun sequencing, but the difference between how we do shotgun and how other technologies shotgun is that we can do CCS sequencing, which allows us to get reads up to 10 kb in length that are highly accurate, just as accurate as short read sequences. And I'll go into more detail about that in a moment. And the great thing about this is that those reads, since they are so long and accurate, you can use them directly for gene discovery without doing assembly. And so we like to think of genome assembly then as a third and distinct application that can be run on the Sequel system. So metagenome assembly from long reads allows you to generate new references, metagenome assembled genomes, or MAGs, for microbes that can't be easily cultured. And in addition, you can leverage the epigenetic genomic data that's gathered during the normal course of sequencing to cluster contigs and plasmids that come from the same host organism. So how do we generate high accuracy and long reads at the same time? So the PacBio system works by sequencing these SMRT bells. So a SMRT bell is made by adding these hairpin adapters on either side of a linear piece of DNA. And once that's on the instrument, the enzyme begins to sequence, and it opens that up into a circle. And we can sequence around the circle over and over many, many times. And when you combine each of those subreads, each of those passes, to generate a single consensus sequence, what you get is a read that has very high accuracy, which we call a HiFi read. And as you can see over to the right, it doesn't take that many passes in order for the next accuracy to be very high. And our average read rates on the Sequel II system are up to 90,000 reads and higher. And as a result, we can sequence both 16S or shotgun reads in very long chunks and get to very high accuracy with those reads. So what does that mean for metagenomics? So I'm going to show you some example data that we generated in-house, and then after that, I will go into some use cases showing how scientists have been able to use that data to better understand complex communities. So in this example here, you have 3 different runs showing full-length 16S sequencing on mock communities. So these are mock communities from ATCC that are either staggered or even. And you can see in both cases there's a large number of reads. And once you filter those reads to be greater than 99% accurate or higher, you can see that the average quality value there is very, very high. So for this example, Q42 means 99.99% accurate or higher. And we're able to multiplex at a very high level here with the average number of reads per barcode as you see below or off to the right. The nice thing about our data set is that while of course you do have to do PCR to get the 16S amplicons, once the template is on the instrument, no additional amplification occurs. And what that means is that with PacBio 16S, you're able to fairly faithfully recapitulate the species that are present in your sample. And so here we're comparing on the left the expected values from the MSA 1002 mock community, and on the right, the actual results from PacBio sequencing. And in addition, the results are very consistent. So this is an example of 32 of the 48 runs, all multiplexed together from the MSA 1009 staggered mock community, and you can see that the results are very consistent across all of those wells. Moving on to shotgun performance, you can see that sequencing performance on the Sequel II system, and this is very robust and gives you a lot of data. So these are 9 runs of human fecal samples using our new chemistry, version 2.0 chemistry, which just came out this October. And you can see in this case, you have on average, well over 2 million reads, and each one of those reads is over 8 kb in length on average. Similarly, when you filter at Q20, you see that the average accuracy of those reads is well over 99.9%. And the thing that I always like to point out here is that if you look at this combination of average read length and quality values, this actually outperforms many metagenome assembly quality metrics. So our HiFi reads are already as contiguous as most metagenome assemblies. And you don't have to take my word for it. You can download this example data set yourself by going to our website. So the other thing that I also mentioned that the PacBio system does not do any PCR amplification on the instrument, and that is even more powerful when you look at the results of HiFi sequencing. So in green bars, you see the abundance of the staggered ATCC mock community expected. And then in purple, you have the results from 16S sequencing on the PacBio system. And you'll see that the green and the purple track pretty closely until you get to the lower abundance values. And then you start to see this fall off. But if you look at the abundance calculated from the HiFi shotgun reads, you'll note that the blue bars continue to track well with the green bars all the way across, and that any discrepancies you see do not correlate with the GC abundance. And that's a characteristic of PacBio data that's been shown over and over again in our work in plant genome sequencing, which you may be familiar with. And that quality allows us to pick up regions of the genome, or in this case, the metagenome, that are often missed by technologies that rely on PCR. So what does it mean that we have these long reads that are highly accurate? So it means that you can do gene discovery directly on those reads without having to rely on assembly. So in this example, we took 3 human fecal data sets. We did amino acid prediction and gene finding on these using FragGeneScan. And what you'll note is that in each one of these cases, there was an average of 6 to 8 genes per read. And the advantage of that is that complete genes can best be found even from species that don't have enough coverage for assembly or error correction. And typically when you do assembly or error correction, you need about 30-fold coverage in order to hit that number where you can get to either reasonable contiguity or high accuracy. But in the case of the PacBio data, that's not necessary. And so if you have any data at all from your 1 or 0.1% abundant community member, then chances are you will be able to find intact genes from that data. Another advantage of this is that it means you can use whatever existing NGS tools that you already use in metagenome assemblies without any modification. So on the other hand, if you really do want to assemble, and there's a lot of advantages to assembling even the HiFi reads, it's true, or maybe you specifically are looking to create metagenome assembled genomes, that works really well as well. Here you can see we've now assembled these 3 different samples to get a range of contigs, and that these assemblies have quite high contig N50. You can also further use a binning tool of your choice. For example, the RBS binning service that's available from the Patrick website to generate high quality bins. The interesting thing, if you look at these bins, is that you'll note that when the PacBio data creates a bin, it tends to either be high quality and really blow that out of the park with very high coarse consistency or fine consistency and completeness, or it does not hit it out of the park. And the reason for that in this case is that this particular binning tool relies on the use of reference genomes in order to seed and create those bins and then find additional contigs that have high analogy to that reference bin. That means that if you do not have a reference for your organism in the database that you're using to do this binning, you're not going to get as good statistics, right? And so what you're seeing here is that when PacBio, with PacBio sequencing, if the organisms in your sample that you're looking to bin are in your reference database, it's going to do a very good job of binning those and giving you a very complete and accurate bin to work from. So now I want to shift gears a little bit and talk to you about a few use cases that researchers have published using PacBio. So as Kelly noted, it's fairly recent that the metagenome scientists have been using PacBio to start to do these types of analyses, and that's because the Sequel II system is really the first instrument that has allowed us to provide data at the scale and at the cost needed to approach some of the more complicated metagenome systems that folks that are studying phytobiomes are interested in. So not all of my examples are going to come from soil or plants, but you'll see as I go along that there are specific reasons why I chose these applications and why I think they are harbingers of the impressive things that you will be able to do with environmental samples. So the first example comes from 16S sequencing. So we have a collaborator, Ben Callahan, who's the author of .2 software. And what .2 does is it analyzes 16S sequencing, and it's very popular software for folks that are doing V4 or other shorter 16S amplicons. So if you will bear with me for a very brief biology lesson, every bacteria, of course, has multiple copies of the 16S housekeeping gene, and the exact number of those copies varies by species, and the differences among these copies varies by strain. And what Ben Callahan found is that when he analyzed data from a mock community using .2, our 16S data was able to recover all the 16S communities from community members, not just an average sequence from each community members, but every exact copy. And that these 16S sequences appeared in integer ratios that reflected their copy number in each genome. So for example, Xanthomonas has 4 copies of the 16S gene, and they are all exactly identical, whereas E. coli has 3 copies that are identical, and then 4 additional copies, each of which is slightly different from the other. And using this detailed information, he was actually able to unambiguously assign the strain in some cases where the database had that information. So in this case, he was able to tell that the E. coli strain in this mock community was unambiguously O157H7 based on NCBI blast results. And this was really eye-opening, both for him and for us, as it turns out, because there had been a rather unflattering report. PacBio data was not accurate enough to do 16S resolution. So in this, however, when Ben reanalyzed the data from this publication, what he found is that the thing that the authors had thought was an error was actually ground truth. So in the PacBio data were 5 distinct copies of the 16S gene from Staph aureus, and they were different enough that when you try to average them together, it looks like systematic error. But actually, that was because the short-read genome assembly that was being used as the ground truth contained only 1 of the 5 RNA operons in the Staph aureus genome. And once they had the more highly resolved information for PacBio, it became clear that it was actually the paper that was an error and not the data. And so Ben thinks there's a big opportunity here to further extend 16S software to do tracking of strains by sequencing a 16S gene alone, or tracking the presence or absence or the consistent appearance of different strains in time points from the same sampling location. And so what that means is that basically when you use full-length 16S, this allows you to bring much higher resolution to even complex environmental communities. So this example is from the sequencing project that was done at JGI, looking at the bacterial composition of the Saginaw Lake on the Sunshine Coast of British Columbia. And this is a meromictic lake that's rich in many different species. Meromictic means that this lake has different layers of water which do not ever mix. And so the researchers took water samples from different lake depths and then did full-length sequencing. And then they followed that up by in silico carving out the V4 only sequences and then comparing what level of analyses and conclusions they would be able to draw with both of those data types. And what they found is that when you have full-length sequencing, you are much better able and much more consistently able to classify the species that the organisms that you find down to the genus and even in many cases the species level. Whereas when you use only V4, you can definitely get class and family, but the ability to classify at the genus level or the species level drops off quite a lot. And so, according to the authors, full-length sequences provide a more complete picture of community composition needed to accurately link microbial players with important biogeochemical cycles within a given ecosystem. Here's a more complete picture of the data that they derived. Now, what you can see here is that along the bottom, there are different phyla that are listed. And then across the top, in each case, you have the analysis that could be done with V4 only, followed by, in a similar color, the analysis that can be done with full length. And so each one of these is a pair of data sets. And what you'll note is that with the long-read sequencing, you were able to do unambiguous assignment at the genus level in many cases, whereas if you look at the examples that are boxed in gray, if you had only the V4, that data did not make it onto this table because it did not include genus level information. And you can see that multiple phyla were completely missed if you rely only on the shorter sequences. And you can imagine how that might impact your ability to really understand who the important players are and what keystone species are present at each layer of that lake. So I'm going to pivot now and move on to some examples of our HiFi sequencing to do metagenome discovery of both genes and then metagenome assembled genomes. So this is a really terrific collaboration that we did with the folks at Second Genome. So Second Genome is a pharmaceutical company who aims to identify novel therapeutics to address unmet needs by doing metagenome mining. And they asked the question, can PacBio sequencing enhance their pipeline for gene discovery if their goal was to identify unique, complete, and error-free gene clusters? So the method that they used for this was to begin with HiFi sequencing, and then to use those HiFi reads first to predict genes directly, but then to also assemble those reads into contigs. They then dereplicate the genes that they find from the reads and map those onto the contigs that come from the assembly. And in that way, they can do additional gene finding on the contigs and understand the relationships among those genes. They also did some metagenome assembly. So this is an example of the assembly graph that you see, the string graph. And so just to help you interpret this a little bit, every time there's a change in color in this string graph, you're moving to a different organism. And every time there's one of these intersections here, these star points, that means that the assembly graph cannot be further disambiguated, and that's where the assembly will be broken and shorter reads will be output. So the interesting thing about this assembly, this is of a soil sample, is you'll note that in many cases, these contigs comprise the entirety or the near entirety of the species present in the sample. And in a few cases, for example, this Pseudomonas and this Acinetobacter, we were able to recover complete genomes complete closed genomes from this sample. Interestingly, in many cases, we were also able to recover plasmid sequences, and in a few cases, these plasmids were also closed. So the main finding from this collaboration was that the HiFi reads are a really great tool for doing gene discovery. And since they are both long and highly accurate, you don't actually have to do assembly in order to find those genes. Different gene finding tools, FragGeneScan and Prodigal, to do gene discovery on their sample. And what the take-home message here is that when you're determining community composition, so for example, through 16S sequencing, the number of observations is what's going to drive saturation. But data contiguity is a more important driver than raw bases when you're doing gene finding. And that long reads allow the recovery of intact gene clusters that are going to produce bioactive small molecules, for example, antibiotic resistance genes or antibiotics, even without assembly. And on top of that, the folks at Second Genome told us that not only were long reads more effective in finding genes, it was actually less expensive. And so here you have a table showing the cost normalized number of genes that can be recovered using either short or long read technology using one of the two gene finding tools that they found. And what you'll note is that HiFi data recovers on average about twice as many genes as short read technology is able to do, even though more short read data was collected than long read data. So I'm going to share now a second example of gene finding and also metagenome assembly. So this relates to the cattle-cow rumen. So this is perhaps a little bit off the field from phytobiome research, but I think that you'll see why I brought it up as a very interesting example of what can be done to understand complex communities. So in this project, the researchers were interested in understanding how short and long read technologies can compare in assembling the cow rumen microbiome. And in addition, how does adding high-speed linkage data add value to that assembly? And in this example, they used Illumina NextSeq sequencing, and then they used data from the PacBio RS2 and the Sequel instrument. So the Sequel is our previous instrument, which was able to sequence 1 million observations at a time, whereas the Sequel II instrument is able to generate 8 times as much data with 8 million sequencing wells. And what you'll see here is that, as with genomes, long-read data produces more contiguous assemblies despite lower coverage. And so here you have the amount of data that was collected from Illumina versus PacBio. And what you'll note is the contiguity of the PacBio assembly is higher than that of the Illumina assembly. And that the contigs from the long-read assembly were on average 7-fold longer. And again, as we saw with the Second Genome data, this really increases your power for gene discovery. So as part of this study, they looked at the open reading frames that they could find in both assemblies across a range of gene families. And what they found is that despite lower coverage, PacBio recovered significantly more antimicrobial resistant genes in the cow rumen than the short read assembly was able to do. And again, this is because gene finding really depends on contiguity. And these results actually contradict previous work that concluded that feeding animals antibiotic concentrates did not lead to resistance. As you can see, the incidence of tetracycline resistance in the cow rumen is actually quite high if you use the right tool for the job. And then finally, when you add Hi-C into this equation, the authors were able to discover 188 novel viral host associations in the rumen microbial community. So as Kelly noted, metagenomes are not just bacteria, they are also viruses and fungi as well. And Adding this additional layer of data along with long-read sequencing allowed them to start to piece together how those viruses and the bacteria in that community interacted with each other. So in conclusion, the authors were able to find that layering in many different types of data and compositional methods worked in a synergistic way to improve the characterization of the highly complex community. So for example, 16S is a really great way to see very rare species using a high throughput method, but using a full length method, you're able to better resolve the community members at the species level. And then using genome sequencing, you can collect large amounts of data using the short read method, which is, again, adds to your ability to do species identification. But using long read data allows you to get better assemblies, which perform better with gene finding and gene discovery. And then finally, they saw that adding proximity ligation can begin to link together all the extra chromosomal DNA that's occurring in a metagenome and start to untangle those important relationships. So the last topic I wanted to touch on here is metagenome assembled genomes. So PacBio metagenome data is able to provide reference quality assemblies of strains. So again, this is a fairly new area for us. Now that we have the Sequel II system, we're able to generate the amount of data that's really required to do a good job with this. But we have already a few use cases of customers doing this. So the first use case was presented at ASM this past year, and it's an example of a researcher who wanted to explore B. breve present in the preterm neonate gut microbiome samples, because this bacteria has been shown to be predictive of a good outcome with preemies. B. breve is unique in that it is one of the species that is best able to convert human milk oligosaccharides into short-chain fatty acids, which basically feeds the gut microbiome. And what they were able to do is to assemble 3 distinct strains of B. breve from the metagenome data. And this was really significant because prior to this effort, there was not actually a reference available for B. breve, and now there are 3. And here's another example that I think is perhaps more pertinent to phytobiome research. This is a case where it was found that you were able to do complete genome assembly of Wolbachia species by sequencing the host Drosophila melanogaster in this case. And so to do proof of concept for this, the researchers took 3 geographically dispersed strains of Drosophila and then sequenced those organisms, and they were able to develop completely closed genomes of 3 distinct Wolbachia strains. And each of these strains had just one contig and was a closed genome, high quality assembly. And the advantage of this is that Wolbachia is, of course, extremely difficult to culture. It's impossible to culture. And in this way, you're able to do assemblies without any kind of experimental enrichment of the symbiont DNA or computational read filtering before you attempt assembly. You just assemble it all together with Canoe in this case, and it falls right out. So this is, of course, really exciting because endosymbionts are, of course, extremely important to complex systems. Not only are endosymbionts present in a wide range of insect hosts, but they also are present, of course, in things like root nodules and other parts of plants. And this seems like a very promising way to start to get high quality genomes of these when they cannot be cultured. Okay, so I'm going to pivot in a slightly different direction now and talk to you about microbial whole genome assembly of isolates. When you do a lot of metagenome sequencing, often it becomes the case that there are a few members of that community that you would really like to get closed genomes of or to study in more detail. And so one way to do that is, if it's possible, to culture and isolate those and sequence them. So then the question is, how much sequencing is enough, or how good of a genome is good enough? And I think until fairly recently, most folks would say that a draft assembly is sufficient, or at the very least, was all that was affordable. And so I want to try to persuade you that we are now entering a new era of sequencing where the high cost of having a draft genome now exceeds the cost of finishing a genome with long-read sequencing technology. So this is a recent paper that I read from 2018, wherein researchers were interested in understanding specifically what is the difference between what you can tell from a draft versus a closed genome. And so they went into NCBI and they decided to look at Bacteroides genomes because there were so many in there, and because there's so many Bacteroides in systems that humans care about. And they decided to look specifically at Porphyromonas gingivalis strains. And what they found is they took 3 of these draft strains, and then they ordered those strains and then resequenced them using PacBio to get closed genomes. And then they did annotations for both the draft and the closed genome, and they compared the results. And what they found is when they were attempting to assemble the draft genome to short read data, what they found is that the results were widely variable depending on what assembly tool you use, and that tools that favored larger contigs were actually generating incorrect assemblies that had frankenproteins in them, and in which the genes were not in the correct synteny, and that oftentimes the genomes assemblies that were better quality or more accurate had more contigs. And so this is, of course, a risk when all you have is a draft genome. You don't really know which of those drafts that you generate is the most accurate for your purpose. And what they found, in addition, was that these draft genome misassemblies and truncations significantly compromised gene finding, and that the draft genomes tended to have more genes, actually, than the closed genomes, because those genes were chopped into pieces and were not recognized as such. And then, in addition, when you have the draft genome, a lot more of the discovered genomes were in the hypothetical category, and their function could not be guessed at by looking at the various domains that were present in the protein. Whereas with the closed genome, you're more likely to find complete genes and then be able to recognize the potential function of that gene. And also, you're far more able to distinguish active genes from pseudogenes, which is often blurred or incorrectly annotated in the draft genomes. So in summary, the authors felt that closed genomes are really required to study a lot of the things that drive microbial genome sequencing. You need them to accurately understand the metabolic capacity of your genome. You need them to be able to understand small phenotypic differences between strains. You need them to be able to look at horizontal gene transfer and to do comparative genomics in any meaningful way across closely related strains. So the good news is that getting a closed genome is less expensive than it ever has been with the Sequel II platform. And the Sequel II platform is unique in that only PacBio is able to close microbial genomes with continuous accuracy across the entire length of that genome, which is really required for robust genome annotation. So I know a lot of you have probably experimented with using ONT for microbial genome sequencing, and you'll be familiar with the requirement for Illumina data to do error correction in order to improve the gene finding from those genomes. But something which is not perhaps well appreciated is that the reason that Illumina data alone cannot close a genome is that a lot of those reads cannot be uniquely mapped to every area of the genome because of ambiguous mapping. And if the Illumina data cannot be uniquely mapped across the entirety of the genome, that means error correction is not happening all around the entire genome. And there will always be regions of that genome that are not accurate. But with PacBio, you don't actually need to do hybrid sequencing. PacBio alone generates very highly accurate closed genomes. And it has just gotten both less expensive and easier to do than ever before. So just this fall, we have released more adapters to allow you to do higher multiplexing on the Sequel II system. And we have just peeled out microbial assembly from the general larger genome assembly tool and established a dedicated microbial assembly tool in SMRT 8.0 that allows you to do automated push-button single technology assembly. So this assembly pipeline works very well for all classes of bacteria, no matter what the level of genome complexity. And the way it does that is with the addition of a chimera detection and filtering module that goes through the data and removes likely chimeras before assembly is attempted. And then we have added a plasmid sequence and recovery assembly stage, which is something you used to have to do on your own, but now it assembles first the chromosomes and then the plasmids. We've also changed our mapping tool that we use to do polishing to employ a circular aware polishing to make sure that coverage stays high across the entire assembly when polishing. And then finally, we've added a couple goodies that are useful for when you're planning to publish your data. The first is that we do circular rotation around the origin of replication for you. And then we output an annotation for that FASTA file according to NCBI guidelines. So on the Sequel system, you can now multiplex 48 microbes per SMRT cell in one SMRT Cell 8M in a single 15-hour run. And so here's the results of the assembly of 48 different microbes that were multiplexed in one run. So the great thing about this is that when you multiplex at this level, the per sample cost for the reagents is only $70. And I think most people would agree that that is an affordable price. Each one of these microbes will have 200 or 4-fold higher coverage, which really makes for very robust and very accurate assembly. It makes it such that you don't have to worry about variable loading or small differences in coverage across all of the pooled microbes. And then finally, methylation data is automatically connected during sequencing, and we have verified that the methylation pipeline works well on the Sequel II system and can be used to annotate the bacterial motifs that are found in different bacterial species. And then finally, I will note, so this is the number of contigs generated for each one of these species. And of course, when you see a 1, that's obviously hitting it out of the ballpark, but I will point out that some of these genomes actually have more than one bacterial chromosome. So for example, B. Cenocepacia has 3 chromosomes. So any number smaller than 3 is of course the wrong answer. And so all of these assemblies are actually very high quality. So how were we able to improve the robustness of our pipeline? So we're still doing assembly in the same way we always did, by using a seed length cutoff to first assembly, do the assembly using only the very longest reads, and that maximizes chromosomal contiguity. But then we go back and we map all the raw data against that draft assembly, and any reads that don't map, we put in its own bin. And then we attempt to reassemble that bin to recover any plasmids that may be present. In this plasmid assembly stage, there's no seed length cutoff, and we use more relaxed parameters that are chosen in order to capture these smaller genomic sequences. Finally, we have added circular aware mapping, and what this does is it allows reads that map to both ends of the draft assembly are available to do polishing on both parts of that genome. And this is distinct from our Falcon or HCAP4 pipeline, which uses a linear mapping tool, which is more appropriate for genomes that are in fact linear. So circular aware mapping, to visualize that more clearly, enables error correction to happen at both ends of that circular chromosome. Whereas with HCAP mapping, you'll see that the coverage drops off here at the one end because those reads are mapping to the other end of the chromosome, and the mapping tool does not realize that it's actually a circular chromosome. So all of these improvements mean that we're getting more reliable closure of chromosome and plasmids without you having to change or adjust the settings or try different parameters or add any Illumina data in order to do error correction. So these are all bacteria that we had our customers send in, asking them, send us your bad assembly data, things that wouldn't close that you thought should close, and because we want to test it out on our new pipeline. And what we found is that all of these sequences that previously did not close are now closing with a circular genome. And these genomes represent a wide range of genome complexity. In addition, we were able to find complete plasmids from the preponderance of these data sets. There were some examples where we did not have a complete plasmid. So for example, in this Bacillus cereus, the plasmid did come up, but it was not complete. And the reason is because it was linear, and our pipeline assumes that they're circular. So we have a little bit more homework to do there. In addition, in this case, we did not get the Shigella sonnae plasmids because there was in fact no coverage in the data. The plasmid, there was just no plasmid sequence in the data. And the reason for that is because with smaller plasmids, there are some issues that remain with optimizing the sample prep. When you go to shear these small plasmids, oftentimes they don't shear because they're supercoiled, or they shear, but they shear into pieces that are so small that the size selection used for sequencing will remove these, and the data never makes it onto the instrument. However, there are ways to specifically extract plasmids and then add them back in to your sequencing. So if you're willing to do those extra steps or to modify the default protocol, you should be able to capture data from those plasmids as well. So why should this matter for phytobiome researchers? So we have a couple of examples which you might be aware of great assemblies of plant genomes. So this is an example of a genome, Heartmenobacter diazotrophicus, that was shown to promote plant growth under salt stress. And these authors noted that draft genomes with low completeness are not reliable for this purpose. They had found already that this plant, or that this microbe was able to have ACC deaminase activity, which is the main mechanism, and it was the main mechanism for the bacteria to promote plant growth under stress. So ACC deaminase is the precursor to ethylene, which builds up during salt stress. And ACC deaminase shunts this over to an alternative pathway. And so interestingly, with the fully assembled gene, they were able to show that there were no genes that were previously known to do ACC deaminase in this microbe, but there were 8 different coding sequences with that same top domain that were found, and that they are now evaluating for activity. And then a second example that you might be aware of is, of course, Xanthomonas. So Xanthomonas are notoriously hard to close because of the tau effector domains that contain. And these tau proteins are very repetitive. They have large number of tandem repeats, and there can be many of these tau proteins in a single genome. So back in 2015, Bogdanov and Leach, who I understand are both members of the Phytobiome Alliance, developed a modified HCAP3 method to reliably assemble these tau genes, and then to feed those assembled genes back into the assembly pipeline and close Xanthomonas genomes. And then what followed was an enormous wealth of publications sequencing many of these Xanthomonas genomes and connecting their activity to virulence and also to mechanisms of resistance in plants. So I hope I've persuaded you that PacBio is the gold standard in microbial sequencing, which is something you may have already known, but also that it's an exciting new tool for understanding metagenomes. And with that, I'm going to stop and turn it back over to Kelly.
Thank you very much, Meredith. It took me a moment there to find my microphone. We have a couple of questions that have come in. The first one, and you alluded to this during your presentation, but maybe if you could restate it, from a practical standpoint, what is the difference in output for 16S sequencing for the Sequel I versus Sequel II?
Yeah, so the difference in output is enormous. So we found with the Sequel I, there was on order 200,000 to maybe 250,000 high quality filtered reads that would come off of that platform. And with the Sequel II, we're getting in excess of 2 million, often 2.4 million reads. And so that really drives down the price point. If you want to have, let's say, 10,000 reads per sample on the Sequel II instrument, you can multiplex at 48 plex and get well in excess of that 10,000 reads, and that will cost you in reagents about $27 per sample. And if you multiplex at 96 plex, then in that case you will get on average 20 to 30,000 reads, though some of those barcodes may have as few as 10,000. That will cost you $17 per sample for reagent costs only. Obviously, if you go to a service provider, there will be a markup, and pricing is a little bit different in different global regions.
So we have another question, is there any evidence of barcode hopping in the PacBio data?
So we have not seen any evidence of barcode hopping. Our barcoding strategy, and one of the things that we do in our pipeline is that we have expected barcode pairs. So our system requires that you tell it whether you're using symmetric or asymmetric barcodes, and it requires that the barcodes that appear in the data match a list of expected barcode pairs. And if it does not, those are thrown out as part of the filtering process. So yeah, we do not see evidence of barcode hopping once things have been processed through the pipeline.
Great. This next question is, how does PacBio data compare to Oxford Nanopore and more recently developed strand sequencing or strand reading technologies?
Yeah, so I would say that the main difference between PacBio and ONT is that PacBio is capable of producing highly accurate reads, whereas ONT requires error correction with short read technology. So with PacBio, you can use the one technology and get what you need, and with ONT, you have to do assembly in order to get the error correction, to do a lot of the types of downstream analysis that people are interested in. And then you have to layer in two forms of technology to do assembly. And of course, when you start to layer in multiple forms of technology to do assembly, that becomes increasingly both time consuming and also resource consuming. And then, see, I'm not aware of other strand reading technologies. I think, from my perspective, I think Nanopore and PacBio are the two most accessible options for folks who want to do long read sequencing.
Great. And then the last question is, does the microbial assembly pipeline work well with small microbial eukaryotic genomes such as yeast?
Yes, it does. I'm so glad you asked that question. Yes, we also ran yeast genomes through the pipeline and got very good results. We assumed that since we were going to call it microbial pipeline, people would put all microbes into the pipeline, and we wanted to make sure that the results were good, and they are indeed very good.
Great. So thank you very much. That's all the questions that we have at this point. I'd like to thank everyone for participating in the webinar. And if you have, just so that you will know, this will be posted on the Phytobiomes Alliance website, which is phytobiomesalliance.org. It also will be, I believe, on the PacBio website as well. And I'd like to thank the Phytobiomes Alliance sponsors, and also to let you know that there will be a Phytobiomes conference next December, December the 1st through the 4th in 2020 in Denver. And you can find information about that on the Phytobiomes Alliance website or on phytobiomesconference.org. So with that, I'd like to thank Meredith again for a great presentation and for everyone who has participated. So thank you very much.
Thank you, everyone.